Welcome to today's deep dive. We have a really fascinating stack of sources for you today.
We really do.
Yeah, we're looking at a PhD dissertation on fungal taxonomy, a morphological study focusing on Wisconsin fungi, and some detailed field guides on the smooth chanterelle.
Which is a pretty impressive mix of data.
It is. Our mission today is to uncover the hidden, explosive diversity of wild mushrooms hiding right under our noses. We are going to see how modern DNA science is completely rewriting the map of our local forests.
It really is flipping everything we thought we knew upside down.
Okay, let's unpack this. Picture a regular, highly trafficked city park in Wisconsin.
Just your average park.
Exactly. People walking their dogs, jogging past the trees, completely unaware of the, alien-like genetic diversity hiding in the dirt right beneath their sneakers.
Right.
Researchers tested a standard patch of chanterelle mushrooms there and found 3 brand new species growing within just 20 meters of each other.
What's fascinating here is that historically scientists and foragers just lumped all of these together.
Because they looked so similar.
Yeah, to the naked eye, they look roughly the same. So it was assumed they were all just the common European species, Chanterelles siberius.
Ah, I see.
But modern DNA testing proved they aren't just minor regional variations, they are entirely distinct species, localized right there in the Midwest.
That is wild.
We now have the pale ghost chanterelle, officially named Chanterelles phasmatis, and a stunning salmon-colored one called Chanterelles spectaculus, both just hiding right there next to the walking paths.
Here's where it gets really interesting, because this hidden diversity isn't just happening with chanterelles. The sources also dive into the hedgehog mushrooms.
The Hydnum family.
Right. We used to think eastern North America only had 5 species of these. Now they've discovered we actually have 16. How did scientists miss 11 entire species of mushroom for so long?
It really comes down to the technology. The researchers used high-throughput PacBio-DNA sequencing.
Which sounds intense.
It is. Basically, that is a method that allows us to read much longer fragments of DNA than older techniques could handle. It gives us a much clearer, high-definition picture of their genetics.
So the DNA pointed the way first.
Exactly. And once the DNA told scientists these were different species, they went back and looked closer at the physical traits they had been completely ignoring.
And suddenly the differences were obvious.
Yes, these distinct visual quirks suddenly stood out. For example, there's a species called Hydnum albarontiacum that aggressively bruises a bright, rusty orange within minutes of you touching it.
Oh, wow.
And another one, Hydnum alba magnum, literally emerges from the dirt with leaf litter permanently stuck to its cap.
So it's not just an invisible lab discovery. You can actually see the difference in the wild once you know what to look for.
Precisely.
But why does jumping from 5 to 16 species actually matter to anyone outside of a botany lab?
If we connect this to the bigger picture, it completely changes how we view the forest network and even conservation. These different mushroom families, both the hedgehogs and the chanterelles, don't just sit on the forest floor waiting to be picked. They form ectomycorrhizal partnerships.
Okay, a symbiotic handshake.
Right, a handshake between the mushroom and the tree roots. The fungus acts as a massive underground nutritional support network. It actively helps trees like oaks and hickories thrive.
So the forest canopy above absolutely relies on this invisible, highly diverse fungal web below.
Exactly.
That brings up a specific mushroom from the field guides that plays a big role in that network, the beloved smooth chanterelle.
Cantharellus latericius.
Yes, it's famous for smelling incredibly sweet, like apricots, and it has a smooth undersurface instead of the typical ridges.
Yeah.
But if the woods are full of all these genetically distinct species that look nearly identical, is there a danger to people out there trying to forage?
There absolutely is. Foraging based on broad assumptions is incredibly risky.
Because of lookalikes.
Right. That beautiful, smooth chanterelle has a very toxic lookalike known as Omphaliotis ollarius.
And that one is definitely not a chanterelle.
No, unlike the smooth chanterelle, the toxic version has sharp, true gills. And in a rather wild twist of nature, it actually glows in the dark.
Which is amazing, but terrifying if you eat it.
Very much so. During the day, to an untrained eye, they can easily be confused.
So what does this all mean? Always consult a local expert before eating what you find. You never really know what you're picking.
Definitely. This raises an important question. If a casual stroll through a highly trafficked city park can reveal 3 completely undiscovered species of a prized, heavily studied mushroom, How many other common plants or insects in your own backyard are actually undocumented species complexes just waiting for a DNA test?
That is absolutely something for you to mull over the next time you step outside. Thank you so much for joining us on this deep dive, and we will catch you next time.